Donc, c'est ça comme pour le propose. Il y a la gestion. Ça va le tour, là, le fait, ça n'a pas marché, mais là, c'est idéal, il faut et puis du coup, je travaille
On peut mettre peut-être là maintenant.
J'ai pas assez de mots.
Et on peut décocher, non c'est super facile à venir sur le nom similaire.
Ouais, oui, il y a un peu plus de mal.
Sinon, ils n'ont pas de voix. Ah oui, d'accord.
Moi, je me tiens là, j'ai une télécommande.
Alors, du coup, on va te mettre un petit peu plus comme ça.
Ouais.
Ouais, en ligne, j'ai commencé, ouais.
Ah, tu veux dire que tu
Faire les gens. Ah ouais.
Attends, je peux
Okay, good morning, everyone. So it's a pleasure to start this year's Thaler Conference. So we are on the webinar mode. So people on Zoom, it means that you cannot interact directly with the room during the conference, but you can report any technical issue in the chat and of course ask your question in the Q&A tab, and we will allow you to speak directly with the speaker during the discussion session. And so before we start, we just wanted to thank the group of students who participated to this year's organization of Thaler Conference. So Marie Riffis, Gwen Vigo, and Alba Marino. And thanks for organizing this very nice afternoon yesterday with non-permanent researchers. And with that, thanks a lot, Nicolas, for introducing to the speaker.
Hi, everyone. It's my pleasure today to introduce the speaker for the 2023 Thaler Conference. Laurent Duret is working at the University of Lyon, where he did his PhD on vertebrates, genomic evolution of vertebrates, and molecular evolution. Then he did a postdoc in Geneva, in Switzerland, and then he got a senior position in Lyon at the what is now the LBBE, basically. Is interested in the evolution of genomes, both selective forces and non-selective or non-adaptive forces, like gene bias conversion, for example. And he has different projects, some of them on recombination and then its role in the evolution of genomes, and another project on the evolution of genomic architecture and the role of genetic drift, and I think he's going to speak about this today. Thank you, and Laurent, the floor is yours.
Okay, so thanks a lot, Nicolas. Thanks, Elodie, and the other organizers of the organizing committee for the invitation. I'm really glad to be here. A bit stressed, but and I really enjoyed also the discussion we had yesterday with the students and postdoc. It was a very pleasant afternoon. And so today I wanted to share with you some result of the PhD work of Florian Benitier. So Florian is interested in investigating to which extent the variation in intensity of genetic drifts that exist among species, to what extent this variation contribute to differences in pattern of expression in expression processes across metazoans. And today I will focus specifically on his work on alternative splicing. So first, some definition. As you all know, in eukaryotes, protein coding genes are interrupted by introns, and so in the nucleus, they are first transcribed to produce a primary transcript, which is then processed to produce a messenger RNA that will be exported in the nucleus and where it will be translated into protein. And so the maturation of the mRNA involves the excision of introns, so the process of splicing. And this process of splicing requires some signals that are present at the extremities of introns, or a donor splice site and an acceptor splice site. And the process of alternative splicing reflects the fact that a given primary transcript can be matured by different ways. So different alternative splice sites can be used to excise introns, so that in the end you can have one gene that produces several distinct mRNAs through the use of distinct splice sites. And so these distinct mRNAs potentially can produce distinct proteins. And this is a very widespread process. For instance, in the human genome, it's more than 95% of genes that are subject to alternative splicing. And a few years ago, there was an interesting observation that was made that so Chen and colleagues measured the rate of alternative splicing across a wide range of eukaryotes, so more than 100 species. And they looked at the relationship between the rate of alternative splicing and organism complexity. So here in this study, they defined the complexity of an organism as simply the number of different cell types that exist in an organism. And they made this interesting observation that the rate of alternative splicing correlates positively with the number of different cell types in an organism. So they observed a positive correlation between organism complexity and the rate of alternative splicing. Another way to show their result consists in plotting what is the percentage of genes that undergo alternative splicing. And what you can see is that there is quite a variation in this parameter. And what you can see is that in vertebrates, the rate of alternative splicing is higher than in other eukaryotes. And so one issue, though, when you see a result like that, is that there's a strong impact of phylogenetic inertia. So basically, this correlation is mainly driven by the fact that vertebrates have more alternative splicing than other eukaryotes. But still, there is this observation. And so this raises the question of the raison d'être of alternative splicing. Why is this process present? And one explanation is that alternative splicing can contribute to the functional repertoire of genomes because one gene can produce different proteins. And there is clear evidence. We know quite a few cases where one gene produces different proteins, typically expressed in different tissues or at different developmental stages. And these different isoforms have different functions. So it occurs that alternative splicing is used to produce diverse functional proteins. However, like any biological process, the splicing machinery is not 100% accurate. And so a fraction of the splice variants that you can observe in a transcriptome, when you sequence the transcriptome of a cell, a fraction of the variants that you observe correspond in fact to errors of the splicing machinery. And these errors, I call them errors because they are costly for the organism. First, because they represent a waste of resources. But second, also because the expression of these truncated or aberrant mRNAs can lead to the production of truncated proteins that can interfere with the function of the normal protein and therefore can be toxic for the organism. And the fact that errors of the splicing machinery are costly for the organism is somehow demonstrated by the fact that within cells, during some molecular surveillance mechanisms have evolved. Some molecular surveillance mechanisms have evolved that are able to recognize messenger RNAs that are erroneous, so that contain premature termination codons, and to degrade these mRNAs. So one of these mechanisms is the so-called nonsense mediated RNA decay. So it's a molecular machinery that is able to recognize and degrade mRNAs that contain premature termination codons. So the question is that, okay, we observe this variation in the rate of alternative splicing across taxa. Does it reflect variation in the repertoire of functional isoforms or variation in the splicing error rate? And a first point is that there is evidence that in some organisms, typically in humans, a large fraction of the splice variants, in fact, do correspond to erroneous transcripts. And the first evidence comes from proteomic studies. So people looked at what are the proteins expressed by human cells. And what they observed is that among all the splice variants that are annotated in the human genome, less than 1% lead to the production of a substantial amount of protein. I mean, a quantity of protein high enough to be detected by proteomic approaches. So most of the splice variants that are annotated within genomes do not lead to a substantial amount of protein. And using different type of criteria, it has been estimated that more than 70% of splice variants in humans result in fact from errors of the splicing machinery. So what does determine the rate of splice error? So first, it depends on the splicing machinery itself. So in trans, that will affect the overall genome-wide splicing efficiency. But there are also some signals in cis, and typically at the boundaries of introns, you have 4 sites that are strictly required. So in most introns, they start by GT and they end by AG. And these 4 sites are strictly required for the proper splicing of introns. And in addition to these 4 sites, there are about 25 other positions that contribute to the recognition of splice sites. And so if you have suboptimal splice signal, then this leads to decrease the splicing efficiency and therefore to increase the error rate. And what the basic principle of population genetics tell us is that we expect that the quality of splice signal should reflect a balance between selection that tend to favor optimal alleles and mutation and genetic drifts that can lead to the fixation of suboptimal alleles. And so the effect of genetic drift depends on the so-called effective population size. So in species that have a low effective population size, so high drift, you expect selection to be less efficient and therefore to have a lower quality of splice sites. And so you expect more splice errors in those species. And so this type of hypothesis is often called the drift barrier hypothesis. And in fact, they come from the work by Ota, the nearly neutral theory, which stands that the capacity of selection to purge Euclid-deletious deep mutations depends on the intensity of random genetic drift. And so this was rephrased by Mike Lynch more recently, where he says that in fact the effective population size imposes a barrier to the optimization of genetic systems by selection. And so what Mike Lynch proposed and is that the effective population size might, variation in any across species might account for variation in different genomic traits, such as genome size, mutation rates, or others. So these relationships that was observed between the rate of alternative splicing and organism complexities are 2 possible explanations. One is a selectionist interpretation. In fact, this correlation might reflect the fact that the evolution of complex organism was favored by the evolution of alternative splicing. So alternative splicing contributed to the evolution of the functional repertoire, which allowed the evolution of complex organisms. So this is the type of interpretation that was given by Chen and colleagues. But we can also propose an alternative explanation, a neutral interpretation, because those organisms that are said to be complex, in fact, they correspond to large animals. And large animals tend to have small effective population sizes. And therefore, we expect them to have lower efficiency of selection because of the drift barrier, and therefore to have a less optimal splicing node and in the end a higher splice error rate. So this is this hypothesis that we wanted to test, to challenge. Does the rate of alternative splicing vary with any or not? And so to address this question, Florian measured the rate of, analyzed the rate of alternative splicing in metazoans. So which shows metazoans because they span a wide range of effective population sizes. And we have a lot of data, genomic data from these organisms. Because what we need to carry out this study, we need a reference genome, we need gene annotations, transcriptomic data, and Florian collected data sets like that for 53 animal species, so 38 insects and 15 vertebrates. And in total, he analyzed almost 300 and 500 RNA-seq samples. So on average, 66 samples, RNA-seq samples per species. And so what he did is that for each species, he mapped the transcriptomic data on the corresponding reference genome to detect splicing events. And a point to note is that he considered all the splice events that are observed in the transcriptome, not only the ones that are annotated in the genome. And so for each detected intron, he counted how many sequence reads correspond precisely to the splicing of these introns. So which we call N1 here. How many sequence reads correspond to splicing events that overlap this intron, but that have a distinct 5' or 3' end? And also the reads that show no splicing events, that overlap these introns, but without any splicing events. So he did that for every single intron detected in the transcriptome. And so at the end, he measured for each species the distribution of this ratio, N1 over N1 plus N2. And what you can see is that you have a clearly bimodal distribution, which implies that, in fact, most genes have one single major transcript. Okay, and you have, in the end, two classes of introns, those major introns that represent the vast majority of transcript of a given gene, and minor splice variants that are observed in a small fraction of the transcript of each gene. And so to measure the rate of alternative splicing, so for each intron, he focused on major introns, so those ones that correspond to the major transcript, and counted the number of reads that correspond to the precise splicing of these introns and the number of reads corresponding to an alternative splicing of these introns, and computed the RH rate. Yes, and he did that only on introns for which the read coverage was above 10. And he focused on introns that are located within coding regions, because introns, you can find introns in 5' UTRs or in 3' UTRs, also in long non-coding RNA. But those introns are subject to different type of constraints. So we focus specifically on introns that interrupt coding regions. One point that is important to notice is that within a genome, the rate of alternative splicing varies among genes. And notably, we had shown in a previous study that the rate of alternative splicing per intron decreases with gene expression level. Okay, so we have a higher rate of alternative splicing in weakly expressed genes. And also, the rate of alternative splicing varies according to the number of introns per gene. So genes that have more introns, the rate of alternative splicing per intron is lower. So given that the rate of alternative splicing varies among genes, if we want to compare the rate of alternative splicing across species, we have to analyze the same set of genes. And therefore, we focused on a set of orthologous genes that are present in single copy in most animal species. So we used the Busco dataset that contains roughly 1,000 genes. Okay, then for each species, we need to have some proxy of their effective population size. And we don't have a direct estimate of their effective population size. For most species, we don't have polymorphism data. So we relied on two life history traits that are believed to correlate with NE. So we took longevity and body size. So these two life history traits are correlated. And we also used the rate of molecular evolution, so the ratio of non-synonymous over synonymous substitution rate, because this ratio is expected to reflect the efficiency of selection to pure weakly deleterious mutations. So it's an indicator of the efficiency of selection. So it's also an indicator, expected to be an indicator of NE. So we use these three life history traits, and just to show, so these different life history traits are correlated between each other, and DN over DS is also correlated with the life history trait. For instance, here with longevity. So here the correlations are computed using PGLS to take into account phylogenetic inertia. So we have a correlation between these proxies of NE, but as you can see, these correlations are far from perfect. So probably none of these proxies capture perfectly what NE is. So now we have, for each of these 53 species, the average rate of alternative splicing measured on Busco genes. And we have these three proxies of the effective population size. And so we can test the hypothesis, do we see a variation between the rate of alternative splicing and NE? So here is the first, the main result. So what is plotted here along the x-axis is longevity. So each dot corresponds to one species, and we colored species according to the clades from which they come. And on the y-axis, you have the average rate of alternative splicing measured across the Busco genes. And as you can see, there's a significant positive correlation. And overall, we have a fourfold variation in rate of alternative splicing between the dipters and primates. And yes, so what I forgot to mention, so that species with short-lived species are expected to have a large NE, and long-lived species are expected to have a low NE. So this positive correlation implies that the rate of alternative splicing is higher in species that have a low NE. Okay, so we also looked at the two other proxies of NE, body length and DNDS, and they also show a significant positive correlation going in the same direction. One limitation of our study is that to increase the sequencing depth, we used all RNA-seq samples that were available for each species. And so we have very heterogeneous sources of data. So for some species, we have a transcriptome from whole bodies. In others, we have transcriptomes from the head or from a specific organ or a specific tissue at different developmental stages. And so in our dataset, we have a very strong heterogeneity in the source of our transcriptomic data. And it had been shown previously that the rate of alternative splicing can vary across cell types, across tissues, or during development. And so to assess to what extent these variation across tissue could affect our results, what we did is that we used a data set that has been published by the group of Henrik Kestman, where they measured RNA, where they sequenced transcriptomes from the same 7 organs in 7 different vertebrates. So they really focused on homologous organs, and they used the exact same protocol to extract and sequence the transcriptome. So these data sets are really comparable. And as you can see, for a given species, the rate of alternative splicing varies across organs. And typically, in most species, the rate of alternative splicing is higher in testes and in brain, and is lower in liver. So it's a pattern that is shared by these mammals and also in birds. So there is some variation among tissues. However, you can see that the variation across species is higher than the variation among tissues. And we still get this positive correlation between longevity and alternative splicing. Okay, so these observations are consistent with the prediction of the drift barrier hypothesis. So this suggests that most of the variants or the differences that we observe in a rate of alternative splicing across species reflect variation in the rate of splicing errors. But as I told you, we know some clear examples of splice variants that are functional. So there are some functional splice variants. So what about the rate of alternative splicing at these functional splice variants? So the difficulty is to distinguish the splice variants that are functional versus the ones that are erroneous. And so we reasoned that those splice variants whose function is to produce a variant protein, for those ones, we expect them to maintain the reading frame. And we also expect that the functional variants that are functional, they should be relatively abundant. Okay. They should represent a substantial fraction of the transcript produced by a given gene. So to test these predictions, what we did is that we took all the splice variants, and for each splice variant, we compute its relative abundancy, relative to all the transcript of a given gene. And so we binned the splice variants according to their relative abundancy. And for each bin, we computed what is the proportion of these splice variants that maintain the reading frame. And so each curve corresponds to one species, and we highlighted 3 species, so humans in brown, Drosophila melanogaster in red, and honeybee in orange. And what you can see is that for splice variants that have a low abundancy, the proportion of these splice variants that maintain the reading frame is about 33, so about 1/3. So this is what you expect simply by chance. But for those splice variants that are more abundant, you see that the proportion of them the fraction of them that maintain the reading frame tends to increase. So this implies that those splice variants that are relatively abundant, they are enriched in functional splice variants. And so we made 2 row categories of splice variants. Those ones that are rare, and those ones so below 5% that represent less than 5% of the transcript of a given gene. And those the other ones that we qualified as abundant. And so we expect functional variants to be enriched in the set of abundant splice variants. So to further test whether these abundant splice variants are functional, we looked at signature of selections on their splice sites. And for this, we looked at polymorphism data. So in Drosophila, we have polymorphism data, and so we looked at the SNP density in the vicinity of splice sites. So we took all the splice sites of the major of the canonical transcript of each gene, and what is plotted here is the SNP density, 20 base pair upstream the donor splice site and the first 20 base pair of the intron. And then we have the last 20 base pair of the intron and 20 base pair downstream in the following exon. And we computed the SNP density at 3 different codon positions. So in yellow, you see the third codon position, so the ones that is less constrained. And as you can see, there's a high level of diversity within introns and within third codon position, so as expected. And a very strong deficit of polymorphism at the donor and acceptor splice sites, so the GT and AG, as expected, because any mutation at those sites will disrupt the splicing, and therefore will be strongly counter-selective. Okay, so very strong selective pressure on the major splice sites of introns. So now, if we look at minor splice variants, so what we show here on that plot is the SNP density. So for those minor splice variants that have one intron boundary that corresponds to a major intron, and the others that correspond to a minor splice site. And so in green, you have the SNP density at a major splice site, so very low SNP density. And in red, you have the SNP density at alternative donor or acceptor splice sites. So for those ones that are located within introns or within the flanking exons. And as a negative control, we took AG and GT dinucleotides located either within introns or within exons, but that cannot correspond to minor splice signals. And so for rare splice variants, so those ones that contain less functional variants, we see little differences between the SNP density at variant splice site as compared to the controls. But when we look at the abundant splice variants, then you can see that the alternative splice sites have a lower level of polymorphism, so which indicates that they are under purifying selection compared to the controls. So this indicates that yes, the minor splice signals of abundant splice variants are under some functional constraints. Okay, all this to say that, okay, among the splice variants, those ones that are abundant, they are enriched in functional splice variants. And so now we come back to look at the relationship between the rate of alternative splicing and some proxies of NE, but separating those introns that have only rare splice variants, so those ones that are less expected to be functional, and we retrieve this same positive correlation as I showed you before, which is expected because these rare splice variants represent 90 to 95% of the introns. And for the small fraction of abundant splice variants, which represents 5 to 10% of introns, here the correlation goes in the other way around. So this time, this correlation is not significant after accounting for phylogenetic inertia, but what you can see is that species that are more complex, based on the number of cell types, so like vertebrates in brown, they have in fact a lower rate of alternative splicing than less complex organisms like dipters. So it goes in the other direction in the opposite direction than what was proposed by Chen to explain the relationship between organism complexity and the rate of alternative splicing. Yes, so this is basically a summary of what I just said. So when we focus on those introns that have rare splice variants, we see that the rate of alternative splicing increases when the effective population size decreases, which is consistent with the drift barrier hypothesis, so higher splice error rate in species with low NE. And conversely, for major introns that have abundant splice variants, so those ones that are enriched in functional splice variants, we show an opposite trend, and we show no evidence for an increase in the rate of alternative splicing in more complex species. It goes, in fact, the other way around. The drift barrier hypothesis makes an additional prediction, which is that the rate of alternative splicing is expected to vary among genes in a genome. So when we measure the rate of alternative splicing, so what we measure is the total rate of alternative splicing, both the splice errors and the functional splice variants. And those that correspond to splice errors, we expect that the cost of these errors should increase with the level of expression of genes. Okay, see, if you have, let's say, 1% of your transcript that correspond to erroneous transcript, this 1% of error will be more costly in a highly expressed gene than in a weakly expressed gene. And so you expect that highly expressed genes should be under stronger selective pressure to limit the error rate. And therefore, the balance between selection and drift will be shifted toward better splice signals, and you expect a lower error rate in genes that are highly expressed. So the drift barrier prediction is that there should be a negative correlation between the rate of error and the gene expression level. So to test that prediction, we focused on introns with rare splice variants, which represent the vast majority of introns. And yes, we expect a negative correlation. And so to test that prediction, what we do is that we bin genes by expression level. And then for each bin, we compute what is the average rate of alternative splicing. So focusing on introns that have rare splice variants. And as you can see, in humans, there's a negative correlation. So the rate of alternative splicing is lower in highly expressed genes as compared to weakly expressed genes. And in Drosophila, the correlation is not as good, but we see the same trend. And so we did that for the 53 species. And in fact, in 52 out of the 53, we observed a negative correlation between gene expression and the rate of alternative splicing. So it's a very widespread observation. So again, it's consistent with the drift barrier model. In most species, we observe this negative correlation between the rate of alternative splicing and gene expression level. So in the end, how do we interpret the observation? So when you sequence a transcriptome, you observe splice variants, and these splice variants correspond to a mixture of splicing errors and functional splice variants. And what the drift barrier hypothesis predict is that the error rate should vary according to NE. So, for instance, here we just to illustrate this, I sketched. I considered 3 species with high NE, medium NE, or low NE. So in species of high NE, you expect a low rate of splicing errors. In species of low NE, a high rate of splicing error. And then we considered, okay, let's assume that 5% of introns undergo functional alternative splicing. And the rate, let's consider that the rate of alternative splicing for this functional variant is the same across all species. Okay. it says that they represent on average 25% of the transcript. So here I zoomed on the distribution to show the distribution of alternative for these functional splice variants. And so in the end, when we look at all variants, what you see is that the average rate of alternative splicing will vary across species simply because the rate of splicing errors vary across species. So this is what is predicted by this model, this mixture model, and this is what we observed for the rate of alternative splicing versus proxies of any. And then if we distinguish those introns with rare splice variants and those introns with abundant splice variants, what the model predicts is a negative correlation for the first ones and a negative correlation for the other ones. And this fits with our observations. And one last point that I wanted to mention is that I showed you previously that in Drosophila, we see evidence of selective pressure on the alternative splice sites of the abundant splice variant. But when we look at humans, in humans, even among those introns with abundant splice variants, we barely see any evidence of selective pressure on the minor splice signals. So no evidence of selection on minor splice sites in humans, even for the abundant splice variants. So to conclude, the splice variants that you observe in a transcriptome reflect, in fact, a mixture of functional variants, which probably represent a minor fraction of all variants, and splicing errors. And so the functional splice variants represent only a small minority of the variants, and typically we can recognize them because they tend to preserve the reading frame. They are generally relatively abundant, and their splice sites are under purifying selection. But variation in the average rate of alternative splicing that we observe across species, in fact, these variations fit perfectly with the drift barrier hypothesis. So this suggests that most of these variations that we observe reflect variation in the rate of error, of splicing errors across species, and not a higher prevalence of functional alternative splicing in complex organisms, as was proposed before. And so this is consistent with the idea that, in fact, random DT drift sets an upper limit on splicing accuracy. And to finish, I would like to come back on this drift barrier hypothesis that was proposed, that was put forward by Mike Lynch. I think it's a very beautiful model, very insightful. But in the end, there is not that much empirical support. There is now accumulating evidence that the rate of mutation correlates with any, so consistent with the drift barrier. This was proposed initially by the group of Mike Lynch, but two studies were published this year that also support this correlation between mutation rate and any. So the results that I just showed you indicate that the rate of alternative splicing also follows the prediction of the drift barrier. In terms of genome size, it's not so clear. Some studies showed relationship between any and genome size that are consistent with the drift barriers, but other studies failed to retrieve this pattern, so it's not so clear. In terms of the rate of transcription error, here for this, in that case, it does not match with the drift barrier. So species with a low NE do not have a higher transcription error than species with a high NE. So in the end, I think it's still an interesting question to pursue. I mean, to what extent does random genetic drift contribute to variation in genomic architecture and in gene expression processes across the tree of life? I think it's still an open question. And an important point, I think, is that when you look at variation in genome complexity across eukaryotes, it's important to consider the null hypothesis of non-adaptive evolution and not to fall in the trap of the pan-selectionist interpretation. Okay, so thanks for your attention, and thanks again to Florian and Anna Maria and Julia who contributed to all these results. Thank you.
Thank you, Laurent, for this very nice talk. So maybe first we take questions in the room. Are there questions?
Thank you, Laurent. Maybe a very naive question, but wouldn't organism complexity also be related with any like organisms that are larger? Would have be more complex, have more cell types?
Yes.
So how to distinguish between that? It makes it difficult to distinguish between the selectionist and the drift hypothesis, right? If complexity is also
okay. So if according to the selectionist hypothesis that alternative splicing contributed to the evolution of the functional repertoire, to the increase of the functional repertoire that allowed the evolution of complexity, you would expect a higher rate of functional alternative splicing in species with in complex species, and it's not what we observe in invertebrates. The rate of functional splice variance, so the rate of alternative at functional splice variance, at least at the most abundant ones, is in fact lower than what we observe in insects.
I had a related question in your model. In the end, with the mixture, you assume that the rate of functional alternative splicing does not vary.
Yes.
But along with what he's saying, will you expect that it also varies with complexity, as you said before, defined, especially when it's defined as a number of cell types, and then that this is correlated with any, which you said from the beginning, from what we know of the biology of these organisms? Because I have the feeling that towards the end, you're saying most of the variation we see is in this error rate, if we assume that the other one doesn't change.
So
the functional part doesn't change, yes. Right?
So if anything, for the functional parts, I see a trend that is a lower rate of alternative splicing in more complex species as compared to well, in species with a higher number of cell types as compared to more simple species. So it's not significant. So that's why I said, okay, let's take the simplest null hypothesis, which is no variation. And so just a simple model where you assume no variation in the rate of functional splice variant, this mixture model is sufficient to explain the observation. But it can be that in addition, the rate of functional alternative splicing might also decrease in the more complex species and not increase.
I have also a related question, actually. Do you think that the fact that I understand that you had to, of course, select genes that are orthologous and present in all of the species, but you might have biased, you might have selected genes that are highly conserved and housekeeping and maybe also rather highly expressed. And do you think that this might influence these results that you find no relation between complexity and rate of alternative places?
So yes, you're perfectly right. So we focused on Busco genes that are genes that tend to be quite constrained, highly conserved across species. So these are genes that are under stronger selective constraints. And so we also looked at the entire set of genes. This is what I show here. And the trend is a bit weaker, but it's still there. So, so yes, but this is an important control, yes.
Yeah, so I was wondering whether you, did you look at the effect of expression level in abundant variants?
Did we?
So you found a negative effect of expression level?
Oh, yes, yes, we did. And so, interestingly, in Drosophila, among the splice variants that are abundant, you see a positive correlation with expression. So in fact, highly expressed genes in Drosophila, if you focus on the abundant splice variants, you see a higher rate of alternative splicing in highly expressed genes.
So this is also expected by your model.
Are there other questions in the room or online?
So, thank you. So you take longevity as a proxy for population size within a context of global change and biodiversity erosion. The population sizes are decreasing, and how does this ongoing process, of quite recent process, but particularly strong, quite recent, influence your observations?
So I think that the rate of alternative, I mean, the traits that we look at presently reflect relatively long-term evolution. I mean, it's the accumulation of weakly deleterious mutations over, I don't know exactly what evolutionary scale, but probably the order of millions of years. So very recent changes in population sizes would not have time to have left an imprint on the mutational load of a species. There was a question.
So I wanted to ask you about the fact that you have rarer, so you have the functional variants are rarer in more complex organisms, if I understand you correctly. But is there, like, for example, you have like distribution of mutation effects generally. So is there you have the frequency of the functional and non-functional types, but do you have a distribution of effects, fitness effects of these kind of alternative splicing things, so that and would having even rare but more rare but something with a larger beneficial effect change how you see would that change anything? That's about it.
Yes, I don't know how to answer. In fact, so the main difficulty is to evaluate the functionality of these variants. So all what I can say is that I have clear evidence that the variants that are abundant. So be careful that here when I say variant, I'm not talking about genetic variation. I'm not talking about polymorphism. I'm talking about within an individual, same gene produces different transcripts. And so I have clear evidence that the variants that are more abundant are enriched in functional variants, but I don't have a precise measure of that.
But if you have some kind of a model that represents this, you could actually test out different fitness effects and see whether whether this is sensitive to how sensitive the drift barrier thing is to that.
Okay, so maybe we should discuss about that. Yeah.
I was wondering if these alternative splicing variants can contribute to phenotypic diversity between individuals. Is there any evidence that individuals could vary in the frequency of the various splicing variants, and that could be translated into variation in traits, or we don't know at all.
So first, so if you have genetic polymorphism on the splice sites, this will affect the rate of usage of the alternative splice sites. Yes, and also you can have some plasticity because so there are different factors that contribute to the splicing machinery, and this factor can be expressed at different levels. And so yes, you can have plasticity and genetic diversity. Yeah. And that's a trait that is under selection, yes.
Thank you. We have another question over there.
Laurent, thank you. So I have a question on human. So you show two results, and I have difficulty to reconcile them. So one is when you increase the abundance of the alternative splices, you have less alternative splicing protein that have a nonsense mutation, but yet, so suggesting selection, but yet when you show the polymorphism on the splicing site, you don't see selection.
Yes.
So is there selection or not? Or
yes, so you're perfectly right. So the point is that, so this study where we looked at what is the proportion of splice variants that maintain, that preserve the reading frame, we had to focus on those splice variants where the distance between the major splice sites and the alternative splice site is not too large, to be, so that we can investigate whether they maintain the reading frame. So we did this only on a subset of all splice variants. While when we looked at polymorphism, we include all of them. So I presume that this is the reason. But you're right. The first result showed that there is a fraction of splice variants that are functional.
Thank you.
Hi. Don't Hymenoptera play a special role in your correlations here?
Sorry?
Don't you think that the Hymenoptera are a bit special in your correlations? And could that reflect perhaps the effect of ploidy?
These are the yellow ones. The yellow ones. The yellow ones. Well, no, it's not obvious that they, neither that they drive, no. There is one outlier, which is a leafcutter bee. The one that has the highest rate of alternative splicing, it's a leafcutter bee. But no, I don't think, I mean, the signal is not driven by Hymenoptera.
No, I think they seem to behave differently, I would say. They seem to go contrary to the general.
Yes, we also looked at whether the social or solitary Hymenoptera differ, and we saw no obvious difference. Because we would have expected the social Hymenoptera to have a lower NE, and we don't really see the pattern.
It's because of haplodiploidy, the selection could be happening differently in Hymenoptera. That's what
okay,
I'm alluding to that, I think.
Okay. Yes.
So, Laurent, so you introduced your talk with this story about the number of cell types, but you never consider the number of cell types in your analysis, and because it would be very compelling to control for both variables to see whether accounting for one you get something more or less, or is that for a given level of given number cell type, you got an effect of any, it would go a long way convincing everyone that it's really an effect of the drift barrier, and whether or not there is a slight effect of complexity, for instance, with respect to those frequent variants that are probably functional. But you could have both,
Yes, so we try to cross our data set with the one of Chen, but in fact, in Chen's data set, there were many unicellular eukaryotes. And for most of our species, we don't have any data about the number of cell types. So in the end, we had very few, a limited number of species for which we had both data. But in any case, I mean, clearly the pattern that was published by Chen is mainly driven by the fact that vertebrates have a higher rate of alternative splicing than other species. And what we see here is that, yes, it is true, but it's essentially due to rare splice variants. And when you focus on the abundant splice variant, you see the opposite trend. So it's, I don't see how you can reconcile that with the idea that you have more functional splice variants in more complex organisms.
Yes, it was mentioned before that was a part of those splice variants that are is kind of uncertain, perhaps. I don't know. Well, it's not significantly different. It's flat.
Yes,
so...
yes. But it's not increasing.
Yeah.
Thank you for your presentation. I was wondering if the expression of the splicing variant could vary during the lifetime of the individual. I mean, if it's different when you compare the embryo stage or juvenile or adult.
So the answer is yes. So the rate of alternative splicing varies both among tissues, but also during development, across developmental stages, with aging, in response to stress. So yes, the rate of alternative splicing varies. Yeah.
Are there other questions in the room? Okay.
Maybe then a second one. So you invoked a neutral hypothesis that says indeed that weakly deleterious mutations should be less well purged in small populations, but that so if you compare species at these scales, what do you know about the selection coefficients of these weakly deleterious mutations? Or maybe what's weakly deleterious in non-complex or less complex species is more deleterious in a more complex species. So do you know if that has been tested, or do you have data on that to ascertain that there's no hidden correlation there as well?
No, not yet. So we don't have the distribution of fitness effect on mutation on splice sites. So yes, that would be a point to investigate. Yes. Yeah.
Thank you. Maybe one last question in the room. Okay, so just to let you know, if you're interested in joining Laurent for lunch, we have like a few seats available, so don't hesitate to come down here. Let's thank again Laurent. Thank you all for coming, and the seminar will continue next week. See you then. Bye.